So this is session 2, symptoms and complications of CF. We've got some really interesting presentations coming up. Our first speaker that I'm going to invite to the stage now is Professor Alan Smyth. He was formerly of Nottingham University. Now he's at Queen's University, Belfast, and his presentation is Gut Feelings, Unravelling Gastrointestinal Symptoms in CF. Thanks, Alan.
Thanks for that introduction, Catherine, and I'm delighted to be here to talk about our Strategic Research Centre, gut research advancing a mechanistic and personalised understanding of symptoms, or Grampus CF. And my principal role in the SRC is to think up the acronym, and I'm particularly proud of this one because I think it really works. As Catherine said, I'm currently at Queen's University, Belfast, but I'm spending time at the University of Nottingham to ensure that the SRC progresses. And if you'd like to find out more, do scan on the QR code, which will take you to our web page, which has more information on our SRC. I'm very grateful to the CF Trust for their funding of our SRC. They've been very generous. We've also had support from the Nottingham Biomedical Research Centre and from our own Nottingham University's charities. The SRC brings together 9 academic institutions, small, medium-sized enterprises, and hospital trust CF centres to try and address what we feel is a critical problem for people living with CF. We've had support from the University of Nottingham, who've supported a PhD studentship, and also from Leeds University Hospitals Trust, which has supported a medical research fellow. But I guess it begs the question, why gut research? I'm a respiratory pediatrician. Why would we be interested in these problems? And you'll have seen these slides already in the meeting today. This is the work of the James Lind Alliance Priority Setting Partnership for CF. And back in 2018, we published a top 10 priorities. We have refreshed that following the introduction of CFTR modulators. And one thing that struck me very profoundly is that the gut symptoms experienced by people with CF are a research priority both in 2018 and now more recently in the CFTR modulator era. So clearly this is of enduring importance to our patient community, and that's persuaded me as a respiratory pediatrician to shift my research focus to this area. GI symptoms are experienced by most people with CF, and they tell us that these are principally bloating, flatulence, abdominal pain. These are intrusive daily symptoms. 2/3 of adults with CF miss work because of gut symptoms. 2/3 of children miss school. And not only do our patients experience these intrusive daily symptoms, but in some cases, the same pathology can lead to severe complications. And all the clinicians in the room will be familiar with distal intestinal obstruction syndrome. The latest registry data show that this affects between 4% and 5% of people with CF. Usually, distal intestinal obstruction syndrome requires a hospital admission. Sometimes it leads to surgery, as was the case in the young person whose abdominal X-ray is shown in the picture. And we believe that the pathology behind both daily symptoms and severe complications may be similar, and we think that the point of mischief is the terminal ileum. And we have developed a theoretical framework, a hypothesis, if you like, to explain why these symptoms occur and possibly why DIOS occurs. And this is a vicious circle, starting off with viscous luminal content, partially digested food which is high in fat relating to pancreatic insufficiency. That in turn leads to dysbiosis, and I'll show you some evidence of that in a moment, and what we call fecalization of the small bowel content, which should be liquid. That in turn leads to inflammation of the gut wall, narrowing the lumen further, and leads to a syndrome of stasis and dysmotility, and then the whole syndrome goes around again. So there are a number of testable hypotheses in this proposal, and we've looked at some of those using MRI scanning. On the left-hand side, there is a control participant. On the right, a person with CF. I want you to direct your attention to the area outlined in red in both of those images. I'm not sure how well it projects, but what you'll see on the control image is that within the small intestine, outlined in red, there's relatively smooth, dark, homogeneous appearance. Compare that with the picture on the right, outlined in red, indicated by the arrow, and you can see that this is heterogeneous and it's white, and we know by comparing this with colonic appearances that the content of the small bowel much more resembles that we would expect to find in the colon. We call this fecalization. Not only that, motility is different. We can measure that with MRI, and motility in CF is about 50% of what we see in matched controls. With any of these slides, if you'd like to scan on the QR code, that will take you to the paper for more detail. We can do a number of things with MR as well as look at the content of the gut. And we have a complicated MR protocol where we have the patient fasting, come first thing in the morning, they have their breakfast, we scan them before breakfast, and then we scan them at regular intervals over a 6-hour period. And we can use those scans to measure small bowel water. And the graph here shows the small bowel water in CF versus control participants over a 6-hour period. And what you can see is that at every time point in that 6-hour period, there is more water in the small bowel in the participants with CF. Not only that, we can measure the orocecal transit time. That's the time it takes a meal to get from the mouth to the large intestine. And you can see how that would be an important metric in a condition that's characterized by obstruction to that transit through the small bowel. And the orocecal transit time in people with CF is 2 hours longer than in comparable controls. Some of the data on the bottom come from Ryan Marsh, who I think we're hearing from later in this session. And he's shown that gut microbiota are significantly less diverse in CF. And he has been able to at least with the satellite microbiota, so that's the smaller 25%, show a relationship between that microbiota and MRI metrics, particularly small bowel water and orocecal transit time. And we interpret this as stasis affecting the diversity of the gut microbiome. So if we know all this, if we've been able to explore some of these mechanisms and demonstrate that they're playing an important role in CF, why do we need to do Grampus CF? Well, the issue is that where you're measuring a physiological variable, you need a relatively small number of patients and controls, or people on therapy and people not on therapy, to show a difference. But if you want to show a difference in symptoms, which at the end of the day is what really matters to patients, you need much larger numbers. You need a larger sample size. And so we've designed a different approach in Grampus. We're going to ask CF participants, 300 adults, 50 children, to fill in symptom questionnaires. And we're going to use a statistical method called latent class analysis to study those questionnaires, because we think that it isn't one size fits all. We think that there will be clusters of symptoms, phenotypes if you like. And we hypothesize that each of those phenotypes will have a different mechanism. So it might be predominantly pain and bloating, predominantly constipation, predominantly fatty stool. We don't know yet, but this is what we think we might find. And we will use MRI, our inflammation studies, and our microbiome studies to drill down to the mechanism behind each of those phenotypes, and we will use that to generate candidate therapeutics. And to do that, we need teams. So we have an MRI team led by Luca Marciani, who's a physicist, and Robin Spiller, a gastroenterologist, an inflammation team led by Tanya Monaghan, a microbiome team led by Chris van der Gast in Northumbria, and Dan Peckham, who's over there, and I lead the clinical team. And bringing it all together, we've got Ian Stewart from Imperial, who's a bioinformatician and statistician. Very quickly, we want to measure symptoms better, so we've designed an app which we're trialing at the minute. This is called Tummy Tracker. It asks 10 questions about gut symptoms. It's designed to be completed every day, and you can see that what you get are the questions presented like the petals of a flower, and you swipe out on the petal to indicate how severe that symptom is for you. We focus not only on the symptoms, but also on the disruption to your life caused by those symptoms in any one day. And basically, if the petal is fully filled in, that's a better outcome. If it's not filled in, that's a worse outcome. So far, 132 participants have helped us do this study. We're comparing it with some standard questionnaires. If you're in the room and you've helped with recruitment to CF Tummy Tracker, thank you very much for your support. The coolest thing about an SRC are the early career researchers and watching them develop in their roles. Understanding the excitement of science and the challenges, but also about communication with lay people, about communication with academic groups, and leadership and team skills. Very quickly, we've got Darren at the front, CF dietitian doing a PhD, Arantxa behind him doing an MRI PhD, Charlotte who's inflammation, Jamila who's bioinformatics, Ryan at the back who's a PhD who's progressed into a postdoc through our GIFT program, Hisham at the very back behind Ian, who is in Leeds with Daniel, clinical research fellow, and Ian Stewart, our bioinformatics guy, who is the mother hen who supports our faculty of ECRs. And finally, the senior citizens, myself and Dan Peckham leading clinical, Luca Marciani in the middle at the back, who's MRI, Chris van der Gast from Northumbria, who's microbiome, and Tanya in the blue jacket in the middle, who's the inflammation lead. And of course, I mustn't forget to mention Becky, who's doing Tummy Tracker. So a fantastic team, and when we get to the Q&A session, I'll be delighted to answer any further questions on that.
Thank you very much, Alan. So we're now going to hear about research on preventing bowel cancer in CF. So please welcome Professor Stephen Renshaw from Sheffield University. Thanks, Steve.
Thank you very much. So I can see we have to get Alan's input on our acronym for our Strategic Research Centre. We don't really have one. So colorectal cancer is a problem for all of us. It's the 4th most common cancer in the UK, but it's a particular problem for people with cystic fibrosis. They have an unexplained 5-fold increase in GI cancers, and this rises to over 30-fold in people who've had organ transplants. And there are many potential reasons for this, but it does seem to be a specific effect of the CFTR genotype, because if you knock out the gene specifically in the colon of mice, those mice develop increased tumours at quite dramatic rates. So it does seem to be something very specific about that gene. So my way into this was quite complicated. I'm a respiratory physician. I don't do cystic fibrosis. I'm interested in inflammatory disease of the lung. And about 20 years ago, I set up a new model to understand neutrophil function in vivo, and that model was the zebrafish. So I'll just give you a couple of quick slides on that to explain. So this doesn't actually project very well in here, and I do apologise, but the great thing about the model is it does allow you to see every single inflammatory cell during the whole of inflammation in the whole animal. So that's the head over the left-hand side and the tail over the right-hand side here. And you can see every single neutrophil. So this has been really useful to us to help us understand inflammation. And any model that helps you understand inflammation also helps you understand host-pathogen interaction. We did a lot of work with Staph aureus, and that brought us to the attention of a really talented postdoc called Audrey Bernoux, who was working on Mycobacterium abscessus in cystic fibrosis. And what she showed here in this top right graph here was that the cystic fibrosis zebrafish model had a specific defect in the handling of Mycobacterium abscessus, and this was due to defective oxidative defenses. And so she came to our lab to do a bit of this work and also to try and look at the primary immune response in cystic fibrosis. And the initial observation was that following injury, there is an over-exuberant inflammatory response in cystic fibrosis. So you can see in the graph here of neutrophil numbers and the images on the right-hand side here. And long story short, Audrey was able to show that this over-exuberant inflammatory response was due to the production of hydrogen peroxide by epithelial cells in response to increased intracellular calcium, which stimulates a calcium-sensitive enzyme called DUOX. And the consequence of this is that if you use verapamil and other drugs of this class, which are some of the most widely prescribed drug classes in the world, you can reduce this inflammatory response back to normal levels, shown here in the graph marked C. So, as another aside, one of the really interesting things about this model is that the tail fin regenerates completely within 48 hours, and that is also defective in the cystic fibrosis fish. So there's, as well as being a defect in bacterial handling and in inflammation, there's also a defect in tissue repair. And all of these are completely independent of infection and chronic inflammation, given the very, very early stages of life that we carry out these experiments. Okay, so that's by way of background. So what this tells us that there's an increased calcium flux in epithelial cells, and if this were also true in the gut, this may give us a couple of mechanisms that might explain the increased incidence of colorectal cancer. So calcium is a proliferative signal. It's a proliferative signal that's usually impeded by the NFAT calcineurin pathway. And of course, that's the pathway we inhibit with tacrolimus and things in patients that have transplants. So that may well be a potential motivator for tumorigenesis, this unimpeded proliferative signal in the epithelium. At the same time, this increased calcium drives increased production of reactive oxygen species, which is an initiator of inflammation in itself, which can then go on to cause cancer or promote cancer. But also the reactive oxygen species can directly damage DNA and be mutagenic. So all of this may feed into a single mechanism. So as it happens, we have talks in the flash talks from all of the people working on this in our strategic research centre. So I'm not going to show any of the data particularly, and they're going to show that themselves at the end of this session. But just to outline, Matt Zilbau is a paediatric gastroenterologist in Cambridge, does some nice work on organoids, who's going to be looking at CF organoids and trying to elucidate the mechanism in human tissue. And just by way of illustrating this is data not from Cambridge actually, but from our collaborator Audrey Bernoux, who's now gone back to form our own lab in Montpellier and remains a member of our strategic research centre. Audrey's been doing some collaborative bowel organoid work and shown that curcumin, which is also a calcium antagonist in a slightly different mechanism, but that this protects against the effect of CF on these organoids and protects against mortality. So my own group in Sheffield, we're trying to define the mechanisms of the tumorigenesis in the zebrafish model, and you're going to hear a little bit more about that today. But this is a transgenic zebrafish that expresses a calcium reporter in the bowel, and you can see that following injury, there's a localized flash of calcium signaling in the zebrafish bowel. And if we quantify the resting level of calcium, there is an increase in the cystic fibrosis bowels. The other piece of work done in Sheffield is going to be carried out by Charlotte Moore, who'll be talking about this in more detail, where she's really drilling down in the mouse tumours to look at the molecular mechanisms that underpin this. We're collaborating with a group in Minnesota, the people who did the original cystic fibrosis knockout in the mouse bowel, and they are doing some extensive work looking to test whether the calcium antagonists are able to protect the mice against tumorigenesis. I'm sorry, the title's been cut off here. But again, hopefully we'll be hearing a bit about that today. And then hopefully at the end of the the SRC, we're going to be able to do some human studies in Cambridge. So thank you very much to the Cystic Fibrosis Trust for the funding, which enables us to do this interesting work, and to all our collaborators and colleagues, and thank you to you all for your attention.
Well, thank you very much for that, Steve. Our final speaker before the Q&A session is Professor Daniel Peckham from Leeds University. Daniel.
Well, thank you very much for inviting me to come and give this very brief talk. I'm another respiratory physician who's talking about the gut, but it's very important. And in terms of our SRC, it was SRC 12, and what I'm going to do is focus on the IGLU CF study, which is an observational cohort study of 175 individuals, 4 CF centres, and during this we were picking up CF abdo scores, the gut microbiome, dietary information, clinical data, modulator therapy, as well as blood samples. But there's also another half to the SRC, which was with Andres Flotto and also Ben Marsland, looking at the basic science as well as the mouse model. Now, terminology, just for those who, like us respiratory physicians, when I refer to the microbiota, we're talking about microorganisms. Then, of course, the microbiome is all about the genes. Then we've heard a little bit about metabolomics. And then you'll see in different studies we'll use either whole genome sequencing or we'll use 16S. And it really depends on what the controls were. Now, the intestines is a very, very important part of the regulatory mechanisms of health. And we know there's over 100 trillion microorganisms. And this can be fungi, this can be viruses, bacteria. And it's also very metabolic. It produces thousands of metabolites. And of course, we know that we can have commensals, it can be symbiotic or pathogenic. And of course, you tend to go from the stomach, from aerobics to anaerobics in the large intestine, but it's in the large intestine you have by far the greatest number of bacteria. Now, the way to look at it, for some of you who remember the Clint Eastwood movies, it is the good, bad, and the ugly. And when you go to lunch, be careful what you choose. Because in cystic fibrosis, the real issue, you've heard it, it's dysbiotic, there's increased pro-inflammatory or potential pathogenic taxes, and a reduction in commensals that ferment. So things, for instance, that can create things like butyrate. And it's very exciting. We've heard about cancer, etc. But in that gut, you've got all these organisms, and the outer membrane, for instance, of gram-negatives will be lipopolysaccharide. These cause a super-inflammatory response in airway cells, in monocytes, in individuals with cystic fibrosis. It's hyperresponsive. You've also got the gram-positive coatings. You've got the short-chain fatty acids like butyrate, which is so important. It'll affect NF-kappa B. It'll affect the cytokines. It'll affect the actual barrier. It'll also is very important for colonocytes in terms of actual fodder. And then, of course, you've got the different bacteria affecting the mucus, the barrier, the immune modulation, and even affecting the autonomic system. So the way to look at it in CF, we have an abnormal pro-inflammatory gut microbiota. And there are many reasons for this. It's going to be CFTR, we've just heard in the mouse, its impact in terms of cancer. It's going to be pancreatic malabsorption. It's also going to be all those antibiotics. It's going to be abnormalities in the mucus, and of course things like PPIs. And this is very important. So when you tuck into that big McDonald's hamburger, it's probably going to help drive inflammation. It causes GI symptoms. The actual variation in the different taxes will have a significant impact. It probably predisposes to cancer. It affects our immunity. It will affect obesity. So the extraordinary thing is you can take the microbiota from a fat mouse and put it in a thin mouse, and the thin mouse becomes fat. So it's quite extraordinary. And also it can even affect the psychology. Now, we previously published some of this information showing that CF has features of autoinflammation. So the cells are hyper-responsive to lipopolysaccharides. And what happens is you get an accentuation of inflammation. And the exciting thing is, I mean, this is driven really through sodium transport, potassium efflux, CFTR, and as we've just heard, actually calcium. And we, excitingly, can downregulate that with CFTR modulators. So GI symptoms, really, this is just flash information. So we've already seen from Jane Davis, fantastically elegant presentation. But here you can see the spider plot. You can see that basically what's happening is that ETI therapy, triple therapy, is basically reducing GI symptoms. Not normalizing, very different, variation spectrum, but we are beginning to see significant improvements. Also, diabetes in its own right is associated with symptoms. And what we've shown is that CF diabetes is also associated with increased GI symptoms, particular symptoms associated with type 1 and type 2. Then in terms of BMI, we're all intrigued, we all hear about it, but body mass index goes up. We know that highly effective modulators increase weight. And what's really exciting about our results is actually what we found is, yes, the BMI goes up, but when we look at nutrition, it actually went down. And this is not surprising. This could reflect, for instance, reduced inflammation, reduced infection, which will reduce actually energy requirements. But parallel to this, we did look at mitochondria. And what we found is actually, in these are cell lines, there's two cell lines. There's a CUFI1, the airway epithelial cell line, and then we've got Margarita from Portugal very kindly gave us these fibroblasts, these kidney fibroblasts. And when you have delta F homozygous, you have an increase in oxygen consumption at basal levels and maximum oxygen consumption. And of course, the mitochondria are the batteries, and they're hyperactive. And extraordinary, you can downregulate it with ETI therapy, but also you can downregulate it by inhibiting calcium, which was so interesting. And actually, the mitochondria are driving mitochondrial ROS, which again is driving other cellular aberrations. So now to fecal microbiota very quickly. Here you can see this is a study which we're looking at the alpha diversity, and you can see we're very lucky to be able to have control data. So we've got irritable bowel syndrome, and we've got controls, so we've got at the top, and you can see how different they are from the cystic fibrosis samples. So in the middle too, those are individuals not on ETI therapy. On the right-hand side, you've got individuals pre and post-ETI therapy. And this is the Shannon score, which is basically reflecting the species richness and evenness. So what we've got to be very careful, we don't see much in this, but of course it won't tell us whether, for instance, the pro-inflammatory species are changing and the anti-inflammatory, so the movement. So again, if you look at it this is when we did 16S, V4, and that's because this was linked into a study on cancer, and you can see that over here, the richness and evenness is very different in cystic fibrosis compared to individuals with cancer and controls. And here, this is the beta diversity. This is, if everything was the same, it'd all be on top of each other. But again, you can see how different it is in the green, which is the cystic fibrosis compared to cancer. Now, this is actually looking at our data in correlation with the same methodology with irritable bowel syndrome. And here you can see, if you look at the red dots on the left-hand side, how different. They're right out, totally dysbiotic. And what's really interesting, if you look on the right-hand side, if you can just look at those blue dots, they're moving down towards those of a healthier, but they're still very dysbiotic. But ETI therapy, that is ETI, basically going from the sort of pink to the blue, that's ETI therapy. So we're seeing actual movement, positive movement in the dysbiotic gut in individuals with cystic fibrosis. And if we actually look, we're seeing enterococcus. And I think the enterococcus is most likely due to the fact we're seeing a dramatic reduction in antibiotics. And the enterococcus is probably being driven up by antibiotics. But E. coli is also significantly going up, very pro-inflammatory bacteria, which could well be driving inflammation systemically and even in the lungs. And then on the right-hand side, we're seeing a reduction in Bacteroides. We're also seeing a non-significant in various other commensal bacteria groups, and we've got loads more analysis to do this early days. And also, again, if you look at this data, you can just see how the E. coli is going down and Bacteroides is going up. And we do feel that we've got to be very careful because some of this could be the impact, because we're seeing a drastic drop in antibiotics. So we're actually at the moment analyzing this data. Now, in terms of short-chain fatty acids, again, this is just really, literally, these slides came on Monday, but the preliminary data is that acetic acid levels go down, propionic acid levels go up, but butyrate, which is really, we wanted that to go up, is not really changing. So overall, we're not seeing anything. But very excitingly, we're very lucky to have dietary data. And if you actually look at the percent butyrate producers, those who are on high fiber diet have a greater increase. And that's in totally contrast to protein, suggesting that the low fiber diet is not doing any good for individuals in terms of producing butyrate, which has very, very important protective mechanisms. And so really in this slide, just think of it as, again, just looking at the differences between the CF, those on modulators or not. What we need to do, and you can see on this image here, what we need to do basically is we need to increase the butyrate, short-chain fatty acids, and the way to do that is increase fiber, protein, and we need to reduce fat. And that will probably have a significant impact. And the near penultimate slide, okay, is cancer. This is hard work in 10 minutes. We've also looked at colorectal associated taxes, and what we found is there's a relative increase in abundance in 6 out of the 20 that are recognized to occur in cancer, which we need to further investigate. So in summary, ETI is partially taming gut dysbiosis. Very exciting. Finally, we're seeing something. ETI is an effective anti-inflammatory drug. It improves abdominal symptoms, potential normalization of metabolism, but we really need to do more. This is one of our patients who very kindly went back to the museum to take a photograph pre and post Kaftrio, but diet, exercise really can impact. And finally, just say thank you to all the collaborators. We've got Andres Flotto, Ben Marsland, we've got Marcus, we've got the Leeds TF team, the Birmingham, Manchester Papworks, because you all need to do it together. And especially thanks to Heled, who's the postdoc, and of course Laura, who really has done the vast amount of this work, who's just been awarded her PhD. And thank you very much, everybody.
Thank you very much, Daniel. So Steve and Alan, do you want to come back onto the stage for the Q&A? I'll just explain how the Q&A is going to work. I'm going to alternate between taking questions from those of you in the room and those of you online. So Belinda will be relaying the online delegates' questions to me. And Belinda, I've got a mic for you, so we just need to make sure this mic one is on. And then here in the room, we have got the roving mic. Jackie's just there in the middle of the room, but we've got the catch box, as I showed you earlier. This is a microphone. As I throw it through the air, it won't be switched on. When you catch it, just give it a second, and then you can ask your question. So I think for this session, I'll see if there's any online questions first. Belinda.
I think the person asking this question was making the connection between Steve's talk and the other two talks on the gut symptoms. And the question was really about what do we know about the gut microbiota's use of calcium?
Who'd like to take that one?
Well, I don't need it. I'm exhausted from that 10 minutes. From what I can work out is what do they feel about the role of calcium? Is that what you think?
I think it was more about whether the bugs produce calcium and whether that could be connected to the calcium effects of preventing bowel cancer.
Now, I think the problem, we know, for instance, that CFTR carriers are actually probably at slightly increased risk of bowel cancer. And obviously, we've seen the very elegant stuff in calcium, but also calcium is a very key part of the mechanisms in cystic fibrosis. We always think of chloride, and I think it's more to do with intracellular calcium, which is due to the abnormality in the iron transport and CFTR. So I think that probably calcium itself is not the problem. It's actually the iron transport defect and intracellular calcium being too high. That would be my impression.
So just to follow up that, I would agree, and I guess then the consequence of that is inflammation, which probably then contributes to dysbiosis, I would guess.
Okay, great, thank you. So should we take a question from the room? Does anyone want to put their hand up? Okay, all right, let's give this a go. I think we're going to have to do this in two parts, although Jacqueline's quite close to you, but I'm quite keen to see how this works. So let's go. So I'm going to chuck it to you first, Andres, and then you can throw it back that way.
Hey.
I don't know, where is it going?
Can you put your hand back up, sir? Thank you.
Oh, Kevin.
Mind the camera.
What could possibly go wrong? Hey.
Just speak into it.
Thank you very much, guys. It's not turned on.
It's not, we're not hearing it.
A very loud voice.
We're going to go with the roving mic, sorry.
Oh, it's such fun as well. Thanks very much, guys. Really exciting work, and it's great that we're beginning to get a window into the gut after all these years. The data that you show for patients becoming established on triple modulator therapy, what you're suggesting is a reversion towards normality, really, but many patients describe having some unsettled bowels when they go on to triple therapy, and sometimes abdominal pain. Did you have any insights into that? Often they're transient, but I wondered if you had any insights in the studies you've done on those symptoms.
We have some physiological insights into that, and the data aren't published yet, but we hope to publish them shortly. And what we've done is do MRI scans of the gut of participants both before and during ETI treatment. What has been intriguing is that the changes that we see are not seen immediately. So the most profound changes are seen after 18 months. And what we see is a reversion of orocecal transit time from the prolonged pattern that we saw, and I presented earlier, to a much quicker orocecal transit, so from 6 hours down to about 4 hours. What is perplexing is that small bowel water doesn't seem to diminish. We thought if we had kind of unblocked, partly blocked sink, that the flow would be better. And I think our interpretation is, and every study generates another hypothesis, doesn't it, that we've partly overcome the blockage, but actually we've switched on secretion as well in the gut mucosa, so that we're getting increased secretion and increased water in the lumen of the small bowel, which is increasing the volume of small bowel water. So I think some symptoms may be better, but not all. And I think the challenge for us is to figure out to what extent we're getting normalization through modulator therapy, and then figure out what is still abnormal, and it might be the microbiota are still abnormal, and then what we can do to help normalize that.
Yeah, I mean, I think what's really important is, firstly, it's very variable between individuals. You're not correcting the pancreas in the adults, so you've still got pancreatic malabsorption. The microbiota is not normal by any means. It's definitely still very abnormal. The diet is grossly abnormal. The amount of fiber is still far too low. The butyrate levels are too low. But these are all things that could change. But I think what we need to do, in a way, I suppose, if you look at the diet we've used in the past, that is a pro-inflammatory, to give high fat, high carbs, poorly controlled diabetes is actually probably the worst thing you can do for a gut. But of course we had to do it because that's how we maintain nutrition. Now we've got a real ability to actually start being really sensible. And even if you look at healthy populations, there's a lot of GI symptoms. And so I think that what's really important is that we are improving symptoms, but we're still far away from where we want to get to. But we're beginning to understand how both the physical gut and the physiology, but also how we can change potentially the content. And that's metabolic as well as the different bacteria.
Thank you. Belinda, any further questions online?
So we have a question. Should CF teams or GPs be performing screening for colorectal cancer at a younger age than the general population in CF adults?
Well, we certainly in Leeds, we screen at the age of 40. So yes, absolutely. And we would screen certainly even much earlier than that if it was somebody who was post-lung transplant. So we heard very elegantly that, of course, lung transplantation accentuates the increase in things like bowel cancer, but also other cancers. So one thing I would say that's really important is things like cervical screening, breast screening. Sometimes these get missed, and it's really important to participate in those. And then, of course, we don't know about the FIT test, which is a standard screening. We don't know whether that's sensitive enough in the CF population, but you'll find that a large proportion of units are now going to colonoscopies at the age of 40 or offering it.
Okay, thank you. So we've got a question down here. Jackie's, I know the catch box is now working, but it's miles away. Jackie's got the mic. We'll bring that to you.
Very interesting talks. Sort of going in the future, are there any studies looking at, say, increasing chickpea and lentil and vegetable consumption to try and, now that the triple modulators are sort of improving it, to try and add onto that with the increase in butyrate or the increase in the fibre content?
That's going to be our study. No, absolutely. What we want to do, we are applying, we personally feel that the way forward is the Mediterranean diet. And there's plenty of research on that. There's loads and loads of paper. The problem is you don't really know, in terms of pancreatic insufficiency, what impact that will have. But there's no two ways about it, is that you can modulate health very substantially by changing the diet. And so that's exactly what we want to do. But no, there isn't, to my knowledge, Laura's the world expert at all the papers, but to my knowledge, there's nothing that really answers that question.
Thank you.
So we've got a couple more minutes before the flash poster session, so we could probably take another question. Belinda, anything lined up from the online delegates?
And kind of on a related note to the previous question, the question was, is there any evidence for routine use of friendly probiotics for CF patients for reduction in symptoms, potentially decreasing the risk of GI cancers?
That's a really good question. There is some evidence that probiotics are beneficial in CF, but not necessarily solely with gut symptoms, but also for respiratory health. There's a very good systematic review of that. I don't think we know at all the effect of probiotics, what they might have on subsequent risk of gut cancer. And there's a whole area of prebiotics which we don't know anything about at all because there aren't very good studies in that area. But I'm not the cancer expert, so I'm going to defer to my colleagues on the panel for a comment on that.
I mean, I don't think there's very good data on probiotics. One thing I would say, which is probably a little contentious, is that actually we know, for instance, there are 130 papers talking about the role of CFTR as a gene, as a cancer suppressor gene. And so it is possible, theoretically, the good news is that potentially ETI therapy by correcting CFTR potentially may have an impact. And so I think that's something to think about. So it's quite exciting. But I don't think in terms of probiotics, to my knowledge, that there is any good data.
Okay, we've probably got time for one more question in the room. So anyone want to pop their hand up? Yep, question at the back. We could throw the catch box at them, but Jackie's got her running shoes on. She's getting there first.
I think the box would hit me in the back of the head.
Yeah.
So with these anticipated increases in cancers in the CF community, is it going to be our responsibility to learn about cancers or oncologists' responsibility to learn about CF? Or both?
Well, I certainly as an adult physician, you know, if you asked me 7-8 years ago, is cancer increase in CF, I'd say of course it is. But the problem is, of course, numbers are so small. I think we need to be very vigilant because if for instance, we know in bowel cancer that it occurs 10 years younger than the normal population. It occurs more commonly in females. It tends to be on the right side. And of course, GI symptoms that you get in cystic fibrosis mimic bowel cancer. So I think it's up to us to start looking at, and this is certainly what our group is trying to do in terms of looking at clinically, you know, advisory stuff, what should we be doing, where are we screening, and all these sort of things are going to be really important. And then, of course, linking in with oncologists. But, you know, you'll find, for instance, things like skin cancers increase, you know, significantly. And it's very easy to miss it because somebody's been to a district general hospital and had it removed. So we need to be aware of skin cancer, advice on protection, post-transplant care, things like, you know, cervical screening, making sure women have had vaccinations. All these things we need to become more aware of, and also make sure that engaging with the primary care and engaging with national screening programs, because this is going to be so important for the future to protect individuals.
Okay, great. Well, thank you very much to Alan, Stephen and Daniel. I'm going to invite you to leave the stage now because we're on to the next part of our session, which is our flash posters. So our early career research is going to be coming up. It's going to be a very fast-paced and dynamic session. The early career research has been funded by the Trust, so they've been asked to give a rapid-fire preview of their posters. They have 3 minutes, and 3 slides to encourage you to go and look at their posters. And so if you're online, you've got the tab there, you can have a look at their posters there. If you're here in London, the posters are out in the foyer. Some of the presenters are presenting remotely. That includes our first presenter, Ephraim Westholm, who's in Lund in Sweden. So while we're connecting with Ephraim, I'm going to ask the others to start lining up along this wall here in the order that you're going to be coming on. And just to let you all know, another small change to the programme, poster 2G from Rahul Bhattacharya will be presented at the beginning of session 3. Once Ephraim has finished, then each of the researchers will come on in turn. They'll introduce themselves. They'll be presenting in a rolling fashion. You'll only hear from me if they've gone over their 3 minutes. So, Ephraim, I hope you can hear me okay. Over to you. Thank you.
Hello. Thank you for this opportunity to present my poster online. So, yeah, next slide, please. I'm from Lund University Diabetes Center, and we mainly work on cystic fibrosis-related diabetes. My project is named IGF-BP7, a new player in beta cell function. As we've already heard from our previous speakers, pancreatic disease and exocrine pancreatic insufficiency is very common in people with CF, and insulin-like growth factor binding protein, or IGF-BP7, has been linked to an animal model, the CFTR knockout ferret, which developed exocrine pancreatic insufficiency and CFRD. What's very interesting is that IGF-BP7, when elevated in exocrine pancreatic cells, can bind to insulin and bind to the insulin receptor and modulate the actions of this hormone. And our own data and data from other groups has also shown that IGF-BP7 can be implicated in the development of type 2 diabetes as well. So with that in mind, next slide please, we formed the following hypothesis. We think that crosstalk between pancreatic compartments, exocrine to endocrine, is important in the development of CFRD, and we aim to investigate how IGF-BP7 affects beta cell function and islet function in CFRD. Next slide, please. So in the first panel here, we see that we have human islets from non-diabetic donors treated with IGF-BP7, and in the blue bars, we see treatment with high glucose, and IGF-BP7 can diminish glucose-stimulated insulin secretion. Go back, please. You go back to the results slide, please. You have gone one too far here. Hello? Thank you. In the second panel, we see that IGF-BP7 in a beta cell line can also repress oxygen consumption rate, implicating impaired mitochondrial function, which explains impaired insulin secretion. And in the third panel, we see that when we reverse the phenotype, when knocking down the IGF-BP7 gene, we see improved insulin secretion. Now to the next slide. So in conclusion, we see that IGF-BP7 can cause impaired beta cell function, and our data supports that IGF-BP7 is a negative autocrine regulator of beta cells, and that IGF-BP7 may be implicated in the development of both exocrine pancreatic disease and impairing insulin secretion from islets. Our future plans is that we want to investigate how IGF-BP7 acts on the insulin receptor itself to modulate beta cells, and we believe that modulating the IGF-BP7 binding site on the insulin receptor could be a potential drug target in CFRD and type 2 diabetes. Thank you for your attention. And next speaker, please.
Hello, everyone. My name is Nicole, and I'm working at Newcastle University and with Ephraim in the same SRC. And my project is looking into the islets of Langerhans in CF pancreata. We know that CF is impacting on the pancreas, as up to 85% of people with CF experience pancreatic insufficiency and requiring enzyme supplementation, and up to 50% of adults with CF develop CF-related diabetes, or short CFRD. This shows with impaired insulin secretion and aberrant glucagon secretion during oral glucose tolerance tests. However, even in advanced cases, we still see that islets of Langerhans are present, but we also see that there's a decrease in beta cell mass, which was reported previously in various publications. So there's something going on in the islets in CF. But the detailed pathogenesis of CF-RD remains unclear, where our SRC is working on different angles to understand this better. I used CF tissue, postmortem pancreatic tissue of CF and CF-RD pancreata. We got 19 donors in total from Germany and Exeter. And we see here 2 example images. This pancreas looks very different to normal pancreas. And we see different degrees of acinar atrophy, which is replaced by fibrosis, which we see here in red, or fat cells. However, in the squared boxes, we see islets are still present, and we saw islets in most of the tissue, or in all of the tissue we were investigated. And my project looked even closer into the islets themselves, and I wanted to know if a mesenchymal phenotype is expressed also in the islet cells. Therefore, I used immunofluorescence staining, which you can see here on these example images. You see insulin in red, glucagon in pink. These 2 are islet hormones, and vimentin in green. This is a mesenchymal marker. And on the top row, you see an example of CF pancreas, and on the bottom, a non-CF, non-diabetic control. And we saw the vimentin can be expressed in some alpha cells, which are marked with the glucagon, and we saw this in CF and control pancreata with a high variety. And we think that the CF environment, the highly inflammatory environment of the CF pancreas, can lead to the development of this phenotype and in CF, and the increased collagen density and increased age can lead to this phenotype in control pancreata. And we think that this is further evidence of the exocrine-endocrine signaling in the human pancreas in CF. So if you want to know more about my project, please come and visit me at Poster 2B.
Hi everyone, I'm Ryan, and I'm going to be talking about CFTR modulator therapy and the gut microbiome and what we know as of now. So a bit of background, we know that intestinal dysbiosis is driven primarily by the primary defect of CFTR dysfunction, which leads to accessory organ damage like pancreatic insufficiency, and there's further relationships with the gut transit and function. On top of that, we know that patients are burdened with loads of antibiotics, additional medications, and a high-fat diet, which further compounds and drives dysbiosis. And we're at a stage now where, obviously, we're treating patients with CFTR modulator therapies, older therapies, monotherapies, ivacaftor, dual therapies, Tez-Iva. That sort of, or the results from that are very mixed. It's a mixed bag in terms of how effective they are. So it begs the question, how are more efficacious therapies are going to impact the microbiota? What do they do? And how does this tie in with gut function and patient symptoms? So what did we do? We took some stool from 20 people CF. We extracted the DNA. We extracted the metabolites, namely short-chain fatty acids, analyzed both of those, integrated some patient data, so symptoms and intestinal outcomes via MRI. And we did that baseline all the way through to extended sampling at 17 plus months. And we also compared that with 10 healthy controls from a previous cohort as well. And what did we find in terms of microbiota? Well, the top panels basically show that over time there's not much change in the whole microbiota analysis, but if you look at the middle top panel, the core microbiota, in the extended period, in the far right bar and the third bar, you can see there is an increase in diversity over time. If we compare back to healthies, we still see, as Daniel mentioned earlier, there's still stark differences in that diversity, which we need to address. Likewise, the similarity of patients. As patients are given the therapy over time, their whole microbiota becomes more similar to one another. So there are some sort of changes going on. And again, it's the same story for the core taxa, which are the more common bugs, bacteria. And the satellites are very variable, and that could be the ongoing sort of antibiotics that are present still at this point, although they are reduced. And likewise, again, there are differences back with healthy controls. which are diminished but are still highly significant in terms of differences, which we need to address. In terms of the functionality, the targeted metabolomics, again, short-chain fatty acids don't really differ over this period, and there are differences compared to healthy controls, which persist. However, the acetic, propionic, and butyric, they are quite similar, so there could be some functional redundancy, which we'd like to investigate further. They're the conclusions I've just whizzed through. And future directions, we want to use wider metabolomics, wider metagenomics to understand these functional differences or similarities further. We want to integrate the patient symptom data and the gut function metrics. And we need to understand the impact of antibiotics in this period in modulator therapy. And yeah, that's going to be explored in Grampus CF. Thanks for listening.
Hi, so first of all, I want to start off by saying thank you for allowing me to present a poster today and talk about the use of patient-derived organoids in research. So, as an introduction, one of the main obstacles to drug testing is that 90% of drugs fail clinical trials, as shown in red. This suggests that our traditional models, such as mouse models and cell lines, are failing to recapitulate human physiology and drug responses, causing promising drug targets to fail in clinical trials and not translate into humans. One of the main limitations, although this illustrates many, I believe is that these traditional models fail to capture the diversity in genetics and also clinical presentation of a disease across a population. This makes it difficult to understand the different disease manifestations and how they may overlap between patients or may be unique to subsets of patients, but it also makes it difficult to understand why patients respond differently to the same drug. Secondly, there's a growing push away from using animal models in research due to ethical concerns, practical constraints, and the time-consuming nature of developing a model like a knockout mouse model. As such, the FDA no longer mandate work in mouse models as a prerequisite for drug approval and recognise the need for alternative models such as patient-derived organoids. So in our lab, we generate patient-derived organoids from fresh and mucosal biopsies of the small and large intestines. These models recapitulate the structure, function, and polarity of the tissue of origin. This allows us to look at disease-specific mechanisms and drug responses at a tissue-specific level. This is particularly important when we're looking at effects of drugs, but also toxicity screening, it may be relevant. Secondly, we also have been able to generate a biobank of patient material containing the primary epithelium, the associated organoids, blood samples, microbiota samples from each gut segment, and also the corresponding clinical information. This means that we can make match comparisons, but also run in-depth analysis of various clinical subphenotypes that may exist and may associate with differential response to drug treatments. Finally, organoids retain the heterogeneity in the patient population at both an epigenetic and transcriptional level, and various studies have shown that they can predict drug response in conditions like CF and colorectal cancer. So we've got a real opportunity for personalised treatment therapeutics. So overall, patient-derived organoids provide a promising model for disease research and drug identification, and will hopefully translate into improved patient outcome across a variety of conditions. Thank you for your attention.
Hi everyone, I'm Charlotte O'Riordan-Moore, and I'm a PhD student at the University of Sheffield. So my PhD project is funded by the Cystic Fibrosis Trust, and we work in collaboration with the University of Minnesota Medical School. So my project title is Mechanisms of Tumorigenesis in CFTR-Associated Colorectal Cancer in Mouse Model Systems. Now it's quite a mouthful. What does that actually mean? So it's already been mentioned that cystic fibrosis is linked to increased risk of colorectal cancer development. Now we're trying to figure out why that is, what are the particular characteristics and drivers of colorectal cancer within the cystic fibrosis intestine. So to do this, we're using a mouse model, and our mouse model has CFTR knocked out within the intestinal tissue in order to mimic the cystic fibrosis gut. And it's already been shown by our collaborators at the University of Minnesota that these mice develop tumours at around a 12-month time point. So what we'll be doing at a 12-month time point is dissecting normal and tumour intestinal tissue from these mice, and our collaborators have also been dissecting tissue for organoid culture. So with this tissue, what we'll be doing is extracting RNA, and we've already run some RNA sequencing to try and work out what are the potential gene expression changes that are occurring within the CF intestine of our mouse and the tumours. And then we've also extracted DNA, and we'll be doing some sequencing and various other analyses in order to try pinpoint what are the certain mutational signatures and what are the certain mechanisms of genomic instability within the CF tumours of our mice. And then we also extracted tissue and formalin fixed this tissue. And what we'll be doing with this is looking at the tissue architecture and also looking at changes in cell types within the cystic fibrosis gut and their tumours, and also be having a look at certain protein changes with particular pathways of interest. So what we're hoping to do with all of this information is to better understand and characterise the tumours that are occurring within the cystic fibrosis gut, if these tumours are similar to that occur within the general population, or if there's something different going on within the CF gut, and what is it that's predisposing these patients to colorectal cancer. So I'm not showing any results in this talk, but if you'd like to find out more, I'm at poster 2E. I've got some preliminary RNA-seq data with some interesting results, and also some immunohistochemistry data. Thank you.
Hi, I'm Stone, and I'm using zebrafish to investigate gastrointestinal cancer from cystic fibrosis. So as we've been told, people with cystic fibrosis are at increased risk of gastrointestinal cancers, and part of the story is due to changes in gut lumen chemistry and dysbiosis. And additionally, CFTR acts as a cell-autonomous tumor suppressor in the gut, as shown by mouse transposon mutagenesis studies, for instance. Zebrafish have strikingly similar CFTR to humans, both structurally and biochemically. So zebrafish, by the time they're 5 days post-fertilization, the larvae, which are still tiny and transparent, they have a fully functional gastrointestinal system, and they can feed themselves, but you can also do live imaging using them. And this allows us to look at the earliest initiating stages for gastrointestinal carcinogenesis before the confounding secondary effects due to the pathology. As people saying, calcium participates in intracellular signaling pathways implicated in carcinogenesis. And using a two-part transgenic system we set up to drive GCAMP, which is a calcium-dependent fluorescent protein in the intestinal epithelial cells. And so you see on the left the tiny zebrafish larvae, and you can mount them in agros for spinning disc microscopy, and you can see the GCAMP expression in the gut. It's a bit hard to see; it doesn't project great, and then can pick them out and genotype them by PCR, because we've got a deletion allele for the CFTR that's easy to PCR. You can see that there's a increased calcium, as shown by the GCAMP in the CFTR mutants, and also using made a intestinal epithelial cell specific GFP transgenic to look at proliferation with EDU. Well, thanks, everyone.
Thank you very much to all of our flash presenters. That was fantastic. So thank you to all the speakers from this morning. And just to remind you that all of the sessions are being recorded. They're going to be available to watch again afterwards. We are now heading into our lunch break, thanks to the flash presenters being very flashy. We have slightly more than an hour, but it gives you more time to go and visit their posters and ask some questions. So remember, online you can look at their posters there as well. Before you disappear, I just want to tell you about the poster judging. So we've got a wonderful panel of poster judges. They're going to be reviewing the posters from 1:45 onwards. So presenters, just make sure that you're all at your posters at that point. For online delegates, we have 4 Zoom breakout rooms set up where Ephraim, Rahul and Arena will be waiting and happy to chat through their posters with you. And those of you here in the room, there are QR codes on your tables. And so if you want to ask questions of those poster authors, then you can go into the Zoom rooms and chat with them virtually as well. And the judges will also use those rooms. There is actually also a 4th breakout room for online delegates to join and chat and interact with each other. So do use that if you would like to. I also, before we go, I just want to mention the summer studentship posters. So there's 5 other posters out there. They're numbers SS1 to SS5. And I'm going to just read out the names of the summer students, and I'm going to ask them to stand up or wave. I know I was told that you might be mortified having to do this, but I know the team is really proud of the work that you've done, and I'd like people to know who you are so they can visit your poster. So we've got Jasmine Johnson and Simran Patel, who's just at the back there, Zayu Chen, Patience Sear, hello, and Ariana Archart. Hello, great, thank you. So do have a look at their posters. So the summer students are funded by the Trust to complete a 6 to 8 week placement. It's over the summer, and then it culminates with them presenting the posters of their work at this conference. So thank you very much, everyone. I'm looking forward to seeing you back here after the break for session 3 at 2:30 p.m. Thank you.